make the students obligation to join your meeting through YouTube today. So we're going to expect many different kind of participant from student, graduate student as well. And most of the attendees here are grant recipients, as I mentioned. So these are professors from different universities in Indonesia. Okay.
Sastia-san.
Yes.
So I have one question.
Yes.
So after showing my PowerPoint file on the monitor, I cannot see any chat.
Yes.
So therefore, I cannot confirm the question from the audience.
Yes.
Is it okay?
You don't need to mind. The administrative staff will compile all of the questions and will display it on the monitor during the Q&A. So Sensei, please focus on your talk. You don't mind to ask about the questions, but to save the time, we will try to collect all of the questions even during your talk. So I'm sorry if there are many notification. You can ignore it in the meantime.
Thanks.
Yeah, if you have any more questions, we'll be happy to answer. Is everything okay, Sensei?
That's okay.
Okay.
Thanks.
So just a little bit of background for everyone. The reason why I invite Naga Sensei is also not because he's a brilliant researcher, but also I attended his talk in Osaka University when you deliver the ACS seminar on how to do scientific writing. And I was very impressed at that time. Most of the presenters present already ready-made presentation from the publisher. And the content is quite similar with what we always see. And Nagai Sensei delivered a totally different presentation, and it was actually very impressive and memorable for all of the attendees because the content is very mind-changing and different. And I feel like as a junior researcher, like I'm still a junior faculty member, I feel a lot of benefit I receive from that. And that's the reason why I ask Nagai Sensei to please allocate your time to share with more people. So yeah, we're very, very honored among your very busy schedule. You are able to join us today. Normally it's very difficult to ask. Nagai Sensei is very busy. And also Prof. Oki also allocate his time today to be with us and explain the motivation why we think this kind of event is very important for the development of our nation. So maybe we will start in 1 minute. We're very close to 1:30 in Indonesia, and we have already 125 participants joining. Perhaps more will join along the way. And before we start, I would like to welcome everyone who are just joining. And I will be moderating this talk in English because our speaker is from Japan. And just a little bit of the rundown of today's event. We from the ForMIND Institute are hosting this event in hope that we would feature world-class scientists in order to inspire us, in order for us to learn from them and from their success stories in terms on how they do research and how they can be able to become leaders in their own respective field. And this event is supported by ForMIND, which is a forum for Indonesian young researchers in Indonesia, and the International Association for Indonesian Scholars, I4, and most importantly, our partner from the Minister of Research and Technology, Indonesian government. So now it's just right on time. We are about to start. I would say again, welcome to everyone who are already here with us. Now we have more than 100 participants already joining. And I would like to welcome also Nagai Sensei and Paoki. And we will like to start our talk by please to give 10 minutes time for Prof. Oki Karnarajasa, the Director of Research and Community Service from the Minister of Research and Technology, National Research and Innovation Agency, Indonesian government, to please give your opening remarks. Prof. Oki, time is yours.
Okay, thank you very much, Dr. Sasti. Please allow me to have share my screen. I hope you all will be able to see the.
Yes,
Can you follow my talk?
Yes, perfect.
Okay. Honorable Professor Takeharu of Osaka University, colleague from ForTi and Indonesian Young Scientists Forum, as well as my dearest participant of Indonesian scientists who are joining the 2 days series of online seminar. Please allow me to show a few of slides to give you an idea why this is very important for Indonesia. So my welcoming remark has a title of Research Capacity Building, a challenge on creating an ecosystem for research and innovation. Indonesia is truly a mega diversity country, and it is very diverse in terms of challenges to manage it. For example, as of July 14, the numbers of population of Indonesia is 273 million population. This is number 4, I think, in the world. We have about more than 1,000 tribes, and we speak in more than 500 different languages. And another challenge is that we have 17,500 islands, but only about 8,000 are inhabited, and the remaining nobody there. So, Professor Takeharu, you may rent the island, but you cannot buy the island because there are so many islands unoccupied at the moment. So, another challenge is that we have too many Indonesian universities. There are 4,724 universities, but only 3,250 universities are under the Ministry of Research and Technology. For example, in China, the biggest in terms of population, they have less than 2,400 universities. And when we talk about the numbers of lecturers, my directorate deals with 265,000 lecturers, but only 15% of them holding PhD, which is a big challenge for us. Also, out of 3,250 universities, 70% are private universities, while national state universities is about 27 universities. So you can imagine how challenging it is to manage research and innovation in the university in Indonesia. How the ministry maintain the quality of university based on research performances. So every 3 years, since 12 years ago, we have conducted what we call it clustering of universities based on research performances. And just like your credit card, we have 4 different clusters: platinum, gold, silver, and bronze. And there are 4 different evaluation components to measure the performances of research in Indonesian universities. Number 1 is resources. Number 2, research management. Number 3, which is the biggest part, is research output, including international scientific publication. Last but not least is revenue generating based on research contract as well as research unit activities. So as you can see here, about 90% of Indonesian university, based on the total that participated in our clustering, are belonging to bronze clusters, while the platinum elite just consists of 47 universities. So in that area, we need to strengthen especially the research capacity buildings, especially when we talk about the university residing in Java and non-Java island. So that is why we cannot just provide research scheme focused on excellent research. We need also the research capacity scheme that will allow them to learn and to become better in terms of conducting research. And this clustering has two consequences. Number one is the budget quota, and second one is the ability or the authority to conduct decentralized research budget. When we talk about research budget distribution, actually our national budget is very low, just about 0.25% per GDP, very much less compared to Japan. And we have allocated into three different categories. Number one is nationally competitive one, account for. Second one is decentralized grant, and the last one is top down, which is account for 10% of the total. And when we see the numbers of proposal we received in the last 4 years, you can see very, very big number. Last year, we received 20,000 proposal, and we funded them only 16,000. But 16,000 is not reflecting the actual of the new proposal, because it's a mix between the new proposal and multi-year proposal that's been ongoing proposal. So in total, from 16,000, there are less than 10,000 are dedicated for new proposal in the national level. And out of that number, 30% are dedicated for those coming from the bronze cluster university. I very much believe that in terms of research cycle, that ideas or brilliant ideas would be one of the most important driven the proposal. So as our request today, we would like to see your experience, your success story, Professor Takeharu, in how you develop and formulating ideas that will be formed into proposal that would be a very successful proposal, which is indicated by the research grant that you got, and then how do you maintain the project, and last but not least is to produce output, especially in terms of international publication in high-level journal. So I very much believe that this cycle will be guidance for Indonesian scientists. So in this regard, we look forward to see how do you formulate the ideas to become a good proposal. Another point I would like to highlight is that the track record. In the assessment of the proposal process in our directorate, there are 3 steps that need to be taken into account. Number 1 is track record, number 2 is the quality of proposed project, and number 3 is the proposed budget. So as part of the online seminar series, we would like to see also how other speakers which share their insights and their perspective about developing track record. Asia also very much concerned about women in science, and another talk is scheduled to accommodate this issue, and we try also very hard to accommodate this issue so that more and more Indonesian women to be participated in the sciences. I think that's all for my short welcoming remark. Again, thank you so much for Professor Takeharu for your time. And I do believe that your contribution will be a great significant for the development of research and innovation in Indonesia. Again, thank you so much also for ForTi and Indonesian Young Scientist Forum, and for the participants. Please make use the opportunity. This is a rare, not many people have the opportunity to join the session by Professor Takeharu. So I wish you all the best. Thank you very much.
Thank you very much, Prof. Oki. We really appreciate your kind introduction to our speaker also, and for all of us to learn the whole process and the whole overview on how the ministry manages the research funding and also how to determine the quality of each university and also how to assess the researcher. And before we move to Professor Nagai's talk, which is our main agenda for today, in addition to the opening remarks, I would like to invite all of the participants here to please turn on your camera, because this rare opportunity needs to be documented. And we hope that we will have a short photo session, although it's online, but we feel that we are now together participating in the same spirit and the same place. So I would like to invite everyone to please turn on their video and camera. Okay. And admin from ForMIND Institute will now take your picture on the count of 1, 2, 3. Okay. Do we need to do again? Okay. Maybe one more time. Maybe you can give me your thumb. 3. Okay. Thank you very much. Thank you very much once again, Paoki, for your wonderful remarks. And I hope if you have time, you can also join the session to see Nagai Sensei's talk. And now we would like to welcome our main speaker for today, Nagai Sensei, who is a full professor in the Institute of Scientific and Industrial Research, Sanken, from Osaka University, Japan. And as you have already seen from his short biography, his research interest is on the biophysics, genetical encoded tools of, like, for example, bioluminescent on fluorescent probes, which he will also mention a little bit in his talk today, and the environmental policy and social systems related. So his field can actually be transferred to and applied to many different kind of scientific fields. So not only biology or only physics. So it will be very interesting. And of course, I would like to read his achievement. He has received in 2018 the Osaka Science Prize and published papers in reputable journals such as Nature Papers, PNS, and also Science. So Nagai Sensei, without further ado, I would like to please welcome you to deliver your talk. And your time is 1 hour. And I would like to invite you to deliver your talk. Thank you very much.
All right. Good afternoon, everyone. And also, thank you so much, Sastia-san, for very nice, kind introduction of me. And so my name is Takeharu Nagai from Osaka University, Japan. Today, I would like to introduce how to come up with innovative ideas for breakthrough research. But most of my talk is concerning my research. But I want to introduce what kind of philosophy is existing behind my research. So this is my main topic of this talk today. So first of all, I must thank to the Sastia-san again for giving me this opportunity to introduce my research as well as my philosophy. This is my great honor. And all right, so first I want to introduce myself. So who is Takeharu Nagai? So as Sastia-san introduced, I'm a professor in Osaka University, and my laboratory is challenging to revolutionize the future society with luminescent proteins. My laboratory is developing so many kind of fluorescent protein as well as bioluminescent protein to see what's going on inside the cell by engineering the fluorescent protein or bioluminescent protein. And recently, we are focusing on the development of so many variant of bioluminescent protein to see what's happening inside whole animal body, or we are now developing the bioluminescent plant. And if I have time, I want to introduce our achievement. And we want to apply this technique for a sustainable society in future. And last December, I and my laboratory member visited Indonesia, Lombok Island. So it's a quite beautiful sea. And the aim of the visiting to the Lombok Island was to capture so many bioluminescent or fluorescent coral. And to do this, we visited this institute, Bio-Industrial Route. And with this researcher in this institute, we captured lots of bioluminescent and fluorescent creature. So this is one example. So captured in Lombok. And this has very beautiful pink or magenta color fluorescence, as you can see here. So like this, our laboratory member sometimes go to trip to get the fluorescent bioluminescent creatures, including animal and plant. And after this, our laboratory perform to get the gene responsible for this fluorescence or bioluminescence. After that, our laboratory member engineer the fluorescent bioluminescent protein to make a genetically encoded molecular spy, by which we can see what's going on inside living cell, as shown here. So this slide shows our genetically representative, our genetically encoded tools. So from Pericum to yellow comedium, so these three are were developed when I was postdoc in Atsushi Miyawaki's laboratory in Brain Science Institute, BRIN, in Saitama Prefecture. So I did I could do very nice work when I was postdoc, and my representative work was the development of Venus yellow fluorescent protein, published in Nature Biotechnology in 2002. And so far, about 2,000 times, 2,000 papers were cited my paper, Venus paper. So anyway, so after engineering the fluorescent or bioluminescent protein, so we made the particular sensors or spy, and then we named each spies, such as Pericum, Venus, yellow comedium, gecko, nanolantern, something like that. And after that, we published papers, so many things like this. So this is, again, the representative work of us. So today, I'm going to talk about one episode or several episodes. So this is outline. I want to talk about fluorescent proteins as well as singularity biology. And if I have time, I want to introduce bioluminescent proteins and its application for future society. Then let me introduce this movie taken by my camera about 10 years ago when I visited the aquarium called Camo Kamo Aquarium in Yamagata Prefecture in Japan. So this is very famous light-emitting jellyfish, Aquaria victoria. So as you can see here, so there is a very, very beautiful green fluorescence from the edge of the umbrella of the jellyfish. So I was surprised to see this. Of course, another researcher or general people also fascinate. And among these, the Professor Osamu Shimamura, who also were interested in this fluorescence. Actually, in 1960s, early in 1960s, he captured so many jellyfish, and from the jellyfish, he succeeded the purification of green fluorescent protein, so-called GFP. And he was the first successor of the finding of green fluorescent protein from the jellyfish. So this is a picture of his portrait taken by my camera when I visit his house in 2004. At the time, he demonstrated the brightness of GFP, and he stored GFP in his house's refrigerator, and he demonstrated this one. So as you can see here, the 50 years old green fluorescent protein still emit very bright fluorescence upon UV illumination. And he reported the green fluorescent protein finding in 1962. 30 years later, US researcher Douglas Brasher succeeded the cloning of the gene. And then the Douglas Brasher provided the gene to the other researchers such as Martin Charfi. He is a neuroscientist, and he really wanted to label the certain neuronal cell in the nematode C. elegans. And just introducing the green fluorescent protein gene into the neuronal cell, he found he could make the fluorescence in the certain neuronal cell. So far, many people believe the cofactor is required for green fluorescent protein fluorescence. However, he demonstrated just introducing the gene of GFP is enough to make the fluorescence, as you can see here. And he reported the success of ectopic expression in the nematode C. elegans in the Science in 1994. After that, Roger Chen also was provided the gene from the Douglas Brasher, and he is chemist, not biologist. He engineered the green fluorescent protein gene by introducing the mutation into the gene sequence, and he developed not only cyan or blue fluorescent protein, but also yellow fluorescent protein. Also, he succeeded the development of the so-called fruit series from the another fluorescent protein found in discosoma, coral discosoma. And anyway, he developed so many color variants. So this accelerates the science, particularly in bioscience, medical science, pharmaceutical science. And owing to this, so we can now doing, for example, visualization of gene expression, visualization of protein localization, cell morphology, biochemical reaction. Anyway, so many applications can be done by using GFP or its color variant. That's why they got the Nobel Prize in Chemistry in 2008, as you know. So here, I want to introduce this very memorable picture, again taken by my camera in 2004. So this sea is called Friday Harbor. It is locating close to the Seattle in US. So in 2004, in summer 2004, the green fluorescent protein related meeting was held in this bay. And this was a picture, a group photo. And so you can find 3 great scientists, Osamu Shimamura, Martin Chaufi, and Roger Tsang. So again, this picture was taken in 2004, 4 years before they obtained Nobel Prize. And but they have already became a giant researchers in this field. So what I was impressed was modesty. So although he was, they were become giant researcher at this time. So usually the big scientists tend to stand up or sit down in the center of the group if we when we take a group photo. But as you can see here, all of these giant researchers stand in the edge of the group or behind the group. So I really thought the modesty is quite important to get the Nobel Prize. But at the time, I sit down in the center of the group. So impudently sitting. So, but I think at the time, I thought at the time, the nail that sticks out is hammered in. However, the nail that sticks out too much is not. So in this picture, I was, at the time, I was early 30s, and this was my philosophy. So although getting the Nobel Prize, the modest behavior is important. However, the career up for young researcher, the nails that sticks out too much is important. So because due to the impudentary behavior, which can attract attention too much by the another researchers. So because the nail that sticks out too much catch attention of someone's eye, and which increase the probability of promotion to a next step. Actually, because of this impudent behavior, many people, including Roger Tsang and Osamu Shimamura, caught, memorize me. So in 2012, I was asked by Dr. Osamu Shimamura to chair his lecture at the Nobel Prize Forum held in the University of Tokyo. And actually, I became a coordinator. So there were so many famous researchers in Japan, but among them, I was selected. So because of the my impudent behavior. So he was impressed. So this impudent behavior is quite important, particularly for young researcher, young student. All right, let me introduce the green fluorescent protein structure. So this is a 3D structure of a green fluorescent protein. It is called bit barrel structure. So from here to here, the length is around 4 nanometer, and from here to here to 2.5 nanometer. And this is the chromophore or fluorophore of green fluorescent protein. And this is the amino acid sequence of wild type GFP, which is composed of 238 amino acids from methionine to lysine. Among these, SYG setting, tyrosine, glycine is the origin of chromophore. So this is the chromophore structure of green fluorescent protein. This is the setting, this is tyrosine, and this is the glycine. And there is a pi conjugation from here to here. So this is very important for the fluorescence of green fluorescent protein. So this is fluorescence from the GFP. So as I said to you before, by engineering the amino acid sequence, we can make the cyan fluorescent protein, blue fluorescent protein, yellowish green fluorescent protein. And also other coral or other animal has orange fluorescent protein. And we can make the red fluorescent protein, much more red fluorescent protein. Currently, world trends is moving to the right to make a far-red or infrared fluorescent protein. This is a world trends. But my philosophy is always sometimes opposite. So this is my philosophy. Go opposite stream of world trends. So that means our way is left side, left direction, not right direction. And I asked to my student, Wataru Tomosugi, why don't we make the much more bluer fluorescent protein? And he agreed, totally agreed. And how can we make such bluer fluorescent protein? So there is a hint, tips in this slide. So this is a green fluorescent protein, fluorophore. So this is a side chain. This moiety is derived from the side chain of tyrosine at 65 position. When we change the side amino acid from the tyrosine to tryptophan, we can make cyan fluorescent protein. If we substitute the tyrosine to the histidine, we can make blue fluorescent protein like this. So we must always utilize the aromatic amino acid in the 66th position. And as you may know, we have another aromatic amino acid. I mean, phenylalanine, F, phenylalanine. It's very similar to the tyrosine, but it's oh, very noisy. It's okay. I continue. So there was an O minus or OH here. So what happened if the amino acid at 66 is substituted for phenylalanine? Like this. So usually it tends to rotate this benzyl moiety. So free rotation of benzyl group disrupt the pi conjugation of this with this. So this causes fluorescence loss. So why freely rotate? Because so O minus of this phenol ring makes the hydrogen bonding network with the surrounding amino acids such as 203 threonine or 148 histidine or 205 serine. So no, but if we remove this O minus or OH, the no hydrogen bonding is made. Therefore, this benzyl group start to freely rotate. So this, so I and my student Wataru discussed how to prevent this free rotation. And we decided to randomly mutagenize the 4 amino acids surrounding the, this benzene group to stop the free rotation. And Wataru actually succeeded to stop the free rotation, and then resulting in the successful making of bluer fluorescent protein, here. And he named this artificial blue fluorescent protein, Sirius, after the planet's star Sirius in Canis Major. And this is absorption and emission spectrum of Sirius. As you can see here, both absorption and emission spectrum is the shortest among the fluorescent protein reported so far. The Sirius absorption is at around 350 nanometer and emission peak is 420 nanometer. So this was the renewal of the shortest emission record of fluorescent proteins after an interval of 14 years. BFP was developed in 1995 and the Sirius was developed in 2009. Anyway, 14 years renewal. Therefore, Waters' result could be reported in Nature method. And he succeeded the four-color imaging in the cellular of the cellular compartment, living cellular compartment. I want to introduce another topic. So by using FRET, first resonance energy transfer, we can achieve the color change. So usually the cyan fluorescent protein is excited by violet light to emit cyan color of light. However, if the YFP, yellow fluorescent protein, is in the close vicinity from the CFP, particularly within 10 nanometer, the excited energy of the cyan fluorescent protein can transfer from here to this chrome hole by FRET phenomena, first resonance energy transfer. And then this YFP is excited to emit yellow fluorescence. Instead, the cyan fluorescent protein cannot emit cyan color of fluorescence. And this 10 nanometer distance, less than 10 nanometer distance is quite important to occur FRET because the 10 nanometer is comparable size of the protein. Therefore, we can investigate protein-protein interaction or protein conformational change by using this physical phenomena. So particularly, we use this phenomena to make a functional probe. So let me introduce one example, very famous example of the functional probe. So calcium indicator. So as you may know, the calcium ion is quite important because this function as a second messenger in most of biological phenomena, including fertilization, cell division, differentiation, cell death, secretion of hormone, muscle contraction, neuronal transmission inside brain. All of these phenomena, calcium ions is involved in. So therefore, the visualization of the calcium concentration change as well as calcium dynamics or spatial pattern is quite important. To do this, my mentor, the previous mentor, Atsushi Miyawaki, developed so-called chameleon calcium indicator when he was in Roger Chen's laboratory. So I was in the University of Tokyo, Katsuhiko Mikoshiba's laboratory. At the time, Atsushi Miyawaki was a research associate in his laboratory. And he stayed in Roger Chen's laboratory as a postdoc, kind of postdoc, and he developed this one, chameleon. And in this case, he utilized FRET and between CFP and YFP, and he utilized CAM, calmodulin. This is a calcium ion binding protein, and M13. This is a 26 peptide, amino acid peptide, derived from the myosin light chain kinase calcium calmodulin binding domain, and they tandemly fused CFP, calmodulin, M13, and YFP. Without calcium ion, chromogen M13 shows the extended form as shown here. Therefore, the CFP and YFP distance is very long. So when we excite 440 nanometer, the CFP emit 480 nanometer cyan fluorescence. But if calcium bind to the chromogen, the calcium chromogen makes a compact complex with M13 like this, which bring the CFP in the close vicinity of YFP. Then thread occur from CFP to YFP. Then YFP emit greenish yellow fluorescence. So therefore, we can we can monitor the calcium existence, calcium concentration change by monitoring the color. If we see the yellow color of fluorescence, it means there is a calcium ion in the inside cell. If we see the cyan fluorescence, there is no calcium ion. With this artificial indicator probe, function probe, they succeeded the visualization of calcium oscillation triggered by the histamine addition, as you can see here. Not only cytoplasmic calcium ion, but also nucleus calcium ion dynamics can be visualized by targeting of this indicator into the nucleus by fusion of the nuclear localization signal. Also, they collaborate with Lex Carr in the Script Institute to make a transgenic C. elegans, in which the pharyngeal muscle are expressing the chameleon. And they succeeded the visualization of the calcium up and down during the eating of bacteria. So this is epoch making. But there is a drawback in the original version of chameleon. So when I was in Shimiaki's laboratory, I improved the performance of chameleon by using the circularly permuted yellow fluorescent protein to optimize the FLET efficiency. By changing the this orientation, as you can see here. And finally, I obtained a very nice variant, YC 3.60, which changed its signal upon calcium binding largely. The dynamic range was 560%. At that time, this dynamic range was the world record. And by using this indicator, I could visualize the calcium propagation, calcium wave inside a single cell. As you can see here, this is a real-time movie. So upon ligand application, such as ATP application, gas pressing application, we could see how fast the calcium wave propagate inside cell. And then after I started my laboratory, the Kazu Horikawa joined my laboratory, and he made ultra-sensitive calcium indicator, chameleon nano. In his case, he modified the linker lengths between chromogen and M13 because he found the longer linker sequence gave higher calcium affinity. This is a calcium titration curve, calcium binding curve. So x-axis indicates the calcium concentration, y-axis the change of FLET signal. And a longer linker sequence shift the calcium binding curve to the left. And the magenta color we named YC nano 15 has the very high affinity of to calcium binding. And the calcium binding affinity of this YC nano 15 is 10 times stronger than EDTA. EDTA is a calcium chelator. As most of you know, we biologists often use EDTA to deplete calcium ion inside solution because calcium often function as a cofactor of enzyme or other something. So therefore, we need depletion of calcium ion. But the YC nano 15 is 10 times higher affinity than EDTA. Therefore, we can deplete all of calcium ion if we add YC nano 15 into the media or into the cell. But we succeed the visualization of calcium signaling propagation among the 100,000 of cells. So this is a multicellular signaling. So when we submit the nature method, the one reviewer said to us that this kind of high affinity version of calcium indicator must perturb the cellular homeostasis by deplete out the calcium ion. But we could visualize the signaling because we could reduce the expression or expression amount of YC nano 15. That's why we could visualize a very, very beautiful calcium or signaling propagation pattern. Anyway, we could be able to detect lower calcium dynamics by using chameleon nano. So the study of YC nano told us many people, even in the textbook, say that if we use an ultra high affinity calcium indicator, it must perturb physiological phenomena, but it is not true, I want to say. So therefore, I want to say here, don't believe the common sense. This is important. Okay, again, they are the representative work from our laboratory or when I was postdoc. By application of our genetically encoded indicator, we want to investigate biological phenomena, particularly singularity biology. So let's move on to the next topic. So to going into the main topic of the singularity biology, I want to introduce several important phrases from the very famous researcher or guy. So first one is the Einstein's very famous phrase. So maybe you know this one. If I had an hour to solve a problem and my life depended on the solution, I would spend the first 55 minutes determining the proper question to ask. For once I know the proper question, I could solve the problem in less than 5 minutes. I was impressed after reading this phrase. And also, I was impressed by the Peter Drucker. He is not a research scientist. He is a management consultant. He also said almost same thing like this. Much of the discussion about decision making is focused on solving problems or answering them. This is a long focus. The important thing is not to find the right answer. The key is to find the proper question. Nothing is worse than the right answer to the wrong questions. But many students, many young researchers tend to focus on to the solving the problem, not finding the proper question. That's too bad. So therefore, I always introduce these two important phrases. So I want to say here a tip to, or I cannot say, so I'm sorry. I can.
Your cursor, Sensei?
A tip, all right. A tip to come up with innovative ideas. This is the main topic of my talk. It's for breaking research is making a proper questions that have a potential to sift the paradigm. Oh, doesn't work. Okay. So what is a paradigm shift? So it's a kind of Copernican change, as you know. So Copernican change is very famous. So in the ancient, in the about 15th or 14th century, many people believed geocentrism. So Earth is center, and Sun is sounding, moving. But after Copernican change, the paradigm was totally changed from the geocentrism to heliocentrism. Okay, this is a paradigm shift. Doesn't work. So how can we find a proper question to toward the paradigm shift? So design thinking is quite important. So do you know the meaning of design? Yeah, many people know design. Yeah, design is designing. Designing is design. But I want to emphasize design can split into D and sign. And do you know the Latin meaning of D and sign? I want to introduce here. So that means leave in Latin, leave. And a sign means mark, given shape in Latin. So therefore, the original meaning of design is leaving from your common sense. Okay, leaving from your common sense. So therefore, the purpose of design thinking is break the vein of your thinking. This is quite important for your research or management of group or getting big funding. So to break the vein of your thinking, find and doubt the assumption is important. So there are so many assumptions in the research. So therefore, the continuing the what if is important. What if for example, what if the protein encodes the gene? What if lipid encodes the gene? The common sense is DNA, usually DNA encodes our gene, but we must doubt such common sense and always think, what if? So how to do the design thinking? So this is a framework of design thinking. So first, make a group with less than 6 people. This is quite important. Lots of people is bad to exchange thinking or idea. And second, list as many scientific problems as possible by exchanging the idea or comment or thinking with these 6 people. And please make more than 100 scientific problem. And after that, focus on to the several most important problems. And then think how to solve the problems as many as possible. So to get as many as possible way, we need design thinking. The common sense part of getting the so many idea. Okay, and fifth, the choose the solution and brush up. And then after getting the idea, perform the experiment. So this is how to do the design thinking in the case of research, scientific research. And also there is a very important rule. First, don't criticize other person's ideas. Criticism makes it hard to come up with good ideas. And second, feel free to say anything. Say anything. This is quite important. don't think that people will laugh at you if you say something wrong. Don't think like this. And third, quantity over quality is important. Give as many ideas as you can. And fourth, build on other person's ideas. So connecting different kind of dot is important to make a totally novel ideas. Connecting totally different knot is important. So to do this, build on other person's ideas. So listen to other people's ideas, be inspired by them, make associations, or add your own ideas to other people's idea and express them as new idea. So this is a scenery of design thinking. So I am the leader of one big project, and this project held last summer the international training course of singularity biology. During this training course, we held the design thinking, and very happy. So half of the participants were from abroad. For example, C or her is from abroad, such as England or other countries. So after getting the idea, we must write down, fill in the idea in this post-it. So after getting so many ideas, we compare the ideas each other. So this is design thinking. So after the design thinking, so evaluation of the candidate questions by checking previous literatures, as many as possible, is important. So we may, although we could get the very nice idea, but there was the previous literature. In such case, we move on to the next idea. So therefore, the checking the previous literature as many as possible is quite important. And after that, we can achieve the getting the proper question. And another important point is a proper question must be shown visually to be transmitted the question to other people. This is also important. So let me introduce my case. I get a very big grant twice, for example, singularity biology and spine minority. And both of these projects were based on the proper question. And to visually show the proper question, I made the main visual, as shown here, main visual of spine minority. So in this case, our concept is how randomly moving small number of molecules cooperatively work together inside cell. And another main visual of, in this case, singularity biology is here. So whether the minor and rare cell would be important for biological system than the major and many cells. All right, let me introduce what's singularity biology and what's the proper question in singularity biology. So before that, I would like to summarize my point. So first, read from common sense by design thinking. Second, find out what the assumptions are. And third, doubt whether they are correct or not. And fourth, make hypotheses with new point of view. And fifth, make a main visual that steers imagination. So what is my proper question in singularity biology? So that is whether the minor population of cells, minor, rare population of cells are important for biological system than the major population of cells. So to explain this, I want to introduce these graphs. So these graphs are representative from so many papers. So I picked up so many graphs, for example, this, and maybe you also often see these kind of graph. So for example, this is a velocity of kinesin motor molecule. And this is a summary of the result. Maybe the author, Lu et al. Measured the velocity of kinesin motor on the microtubule at the single cell visualization, single molecule visualization, and put the dot, more than 100 dot, from which they calculate the average of the velocity of kinesin motor, which was about 500 nanometer per sec. And they show this black bar to show the average of the velocity. And they also show the deviation. The deviation is quite small. And then they discuss the kinesin motor's function by using 500 nanometer per sec. But as you can see here, there are so many rare case. So for example, in this case, this kinesin motor has the velocity of 2000 nanometer per sec. It was 4 times faster than the average. So this is rare kinesin motor. But we must discuss this. However, almost all of the case, the author didn't mention about this rare case. So in the case of this, this is also far from the average. This is also far from the average, almost same to the wild type value, even if this has the mutations. So therefore, I want to say, although element, protein, cells, and so on with properties that are far away from the average are very rare, but always found, little is known about them. So another proper question is here. Is the conventional statistical approach applicable to investigate rare elements? Because the conventional research is based on the random sampling. Random sampling is a conventional method to estimate the property of a population. So this is a mother population. We often purify the several sample to measure the property. And after getting the data of the random sampling data, we estimate the property of mother population by using mean value or deviation. This is a conventional way of the science. But in the case of singularity biology, we want to focus onto the very rare case. And it's a very far from the average average. Okay, I want to summarize. So you often see this graph in the paper. So in this case, there is a 100 point, about 100 point of data. And after measuring the gene expression or cellular activity, so many people focus on here. What can we know from this statistical analysis is average description of a phenomena. So this is a conventional way of science. But in our case, we focus onto this minor cell. The minority has been excluded for statistical analysis as an outlier. Therefore, no one knows what happened if this is activated. And but we pay attention to the minority because it might have an important role for singularity-related event. So The singularity is a spatial or temporal specific point at which phenomenon or system and so on drastically changed into different status. So this is a singularity. And by which the status drastically changed from this to this. So the very famous phenomena is Big Bang. So before Big Bang, there is nothing. After Big Bang, the universe is made. The birth of life is also a singularity-related event. Evolution, AI, and a COVID-19 pandemic is also singularity-related event. Also, the social revolution is the singularity-related event, such as French Revolution, Meiji Revolution in Japan. And a common feature of these phenomena is that they are triggered by minor factors or rare events. So our hypothesis is a minor cell makes singularity that causes drastic change of biological system. Okay. So this is an example of phenomenon in which singularity is involved. So in this movie, there are 100,000 of cells. One dot indicates one cell. So this is a dictyostelium discoideum cell. After nutrient starvation, as you can see here, the aggregation stream is made. So very beautiful aggregation stream. But the aggregation stream is toward only one point. So that means there is a leader cell here. Leader recruit other cell by propagating maybe signal. So this is a very, very drastic change from the single to multicellular system. Therefore, this is a singularity-related event. And a layer cell may trigger this. And to visualize this, we made CAMP, cyclic adenosine monophosphate fluorescent probe to find out the leader cell. Because the CAMP can cause aggregation stream. Therefore, we thought the leader cell must secrete lots of cyclic AMP. So for this purpose, my previous member, Kaz Horikawa, he is now a full professor in Tokushima University. So in collaboration with him, we developed the cyclic AMP indicator R-Link. I skipped this, the detail. Anyway, this indicator emits red fluorescence upon cyclic AMP binding. With this indicator, we could do the transcale imaging. So this is the first success of transcale live imaging of cyclic AMP dynamics in dictyostelium aggregation process. So we could do visualization of the cyclic AMP dynamics or concentration at the single cell level, as well as macroscopic cyclic AMP propagation in the 100,000 of cell. Okay. Red color indicates higher cyclic AMP concentration. Then what we found by transcale imaging is there is a 4 category of cell: leader, early follower, late follower, and citizen. Leader first secretes lots of amount of cyclic AMP, and early follower follows the leader's secreted cyclic AMP, and early follower bind the cyclic AMP, and then this signal triggered the secretion of cyclic AMP from the early follower. So this is a relay of cyclic AMP signal. And now leader, after nutrient starvation, about 3 hours after nutrient starvation, leader secretes the lots of cyclic AMP. And this phenomena occurred 10 minutes interval periodically. And about 3 or 4 times oscillation of the cyclic AMP release, the late follower started to follow. Anyway, we counted the number of leader cells, and we found less than 0.1% population become leader. And the early follower population is 0.5%. Late follower is also about 0.5%. The most of cells are categorized into the citizens, 90%. So here I want to say this is citizen cell, and this is leader cell. But if we don't utilize the transcale imaging, we cannot find this minor cell. In addition to this, not only the minor cell, but also the follower function is important to trigger the movement of the citizens. So this is a pulse number of spontaneous pulse release of cyclic MP from leader. So first pulse, so leader, only single leader release the cyclic MP, and maybe 7 or 8 follower dictyostelium followed. And after fourth pulse release, the first late follower, maybe 3 to 4 late follower cell started to follow. And after ninth pulse release of cyclic MP from the leader, the follower number become more than 20. So this is a singularity because after that, the citizen started to follow the aggregation stream because more than 20 cell release of the cyclic MP release from the more than 20 cell is enough to trigger the movement of the citizens. So I emphasize that biochemical method which grind millions of cells cannot find a minority because we can lyse these all of cell. Therefore, we cannot see the property of leader cell by using biochemical method. Imaging a part of a specimen with microscopy must overlook the minority. Also, macroscopic imaging cannot visualize a minority. So these minor cells may be involved in the singularity also in the higher organisms, such as cancer metastasis. As you know, the primary tumor cell produces a circulating tumor cell, which travel around via blood vessel to make a metastatic tumor. But as you may know, the CTC, circulating tumor cell, is quite rare. 1 CTC per 10 billion blood cells. This very rare cell makes metastatic tumor, which trigger the death of organisms. This is quite a drastic system change from living to death. Also, Alzheimer's disease. So, Hiroko Banai is studying the Alzheimer's disease is triggered by the accumulating 2 to 3 cells accumulating abnormally aggregating tau proteins. Tau is a protein associated with the microtubules. So the start point, only 2 to cell abnormally aggregating tau proteins. But at the certain point of age of human, the exponential increase and the spreading of such abnormal cell to the cerebral cortex, which cause loss of neuron from cerebral cortex, which causes brain dysfunction such as Alzheimer's disease. So this is totally drastic change from the normal to the dysfunction triggered by rare cells. So these are just a hypothesis, but we are now investigating these hypothesis is true or not. But we need visualization of CTC cell or abnormal the locus cellular cells, 2 to 3 cells, which has abnormally aggregating tau proteins. So how? So currently we are thinking to utilize bioluminescence. Yes, 8 years ago, our laboratory succeeded the visualization of cancer tissue inside freely moving mouse. And also my previous mentor when I was postdoc, Atsushi Miyawaki, developed very nice bioluminescent protein to visualize the gene expression deep inside body of a monkey, as you can see here. So this kind of experiment visualization cannot be done by using fluorescence imaging. So bioluminescence is quite important. But the transcale imaging of a much more wider area is required because mouse or monkey is much bigger than the dictyostelium aggregation stream. So we need much more bigger transcale imaging system. For this, the transdimensional life imaging division in Osaka University are now developing Amaterasu, multi-scale model analytical tools for everyday activities in singularity. So what is Amaterasu? So this is a conventional microscopy size. This is a scale bar. And our Amaterasu is this big. Inside Amaterasu, there is a big objective lens and also ultra big image sensor. So because this image sensor is too big, the output data is also big. So we need data feasibility technologies such as data compression, data mining, big data transfer, big data storage. So we are now developing such Amaterasu image sensor by using the conventional Japanese technologies. As you may know, the Subaru telescope was constructed on the top of mountain in Hawaii. The diameter of this reflection mirror is 8.2 meter. And this is main optics in which there is a complicated optics for compensation of the how to say air fluctuation, which perturb the higher resolution imaging. Anyway, all of these techniques are based on Japanese technique. So by using this technique to apply the bioimaging, or particularly, we are now applying these technology into the Amaterasu. So unfortunately, the detail of Amaterasu is confidential now, but we have already developed prototype Amaterasu, by which we can do one-shot acquisition of transcale image from subcellular to whole brain image. So this is an example. This is a mouse brain one-shot image, but we could see the detail of hippocampal neuron. So this is a kind of Google Earth-like bioimaging. Now we also performed the visualization of more than I forgot the number of cell. So this is again the dictyostelium discoideum cell. So as you can see here, there are so many cells, and we can easily find the leader cell here. Right? So this is a powerfulness of Amaterasu. So I want to emphasize a paradigm shift of bioimaging with Amaterasu. So if we want to see the cell, we have been always utilize microscope. If we want to see the organ or whole body, we have been always utilizing the microscope. But amateurs can be used for viewing all details without modification. We can see cell as well as tissue, organoid, organ inside whole body. So as you know, this is a very famous equation by which the spatial resolution is defined. 0.61 wavelengths of light divided by numerical aperture of lens. Many people are believing to see the fine structure inside cell, the magnification by microscope is required. But so this is Leidy's definition. There is no parameter of magnification, only wavelengths of light and numerical aperture of lens, only two parameters, not related to the magnification. So that means no magnification is required for viewing the detailed fine structure inside cell. And according to this, we achieved the constructing of amateurs. Time's up, therefore I cannot introduce bioluminescent proteins. So I want to, just a minute. Finally, so today I talked about how to come up with innovative ideas for breakthrough research. So I introduced several things, what is important. So design thinking is important. So finding what is assumption is also important to obtain what is a proper question. And I demonstrate the proper question obtained by my own self by introducing what is singularity biology. And I want to close my talk by take-home message from the George Whiteside. He is a professor in Harvard University. So final message from me is from George Whiteside. He said, if the research that we do does not change the way people think, the project is a failure. So this is one of my favorite phrases. So according to this phrase, I want to continue my research by which so many people change the way they think or change the paradigm of them. That's all from me. So finally, I want to acknowledge my laboratory members as well as so many funding and also all of collaborators. That's all.
Okay, thank you very much, Nagai Sensei. Very impressive talk. And I would like to invite all of the participants to please unmute your speaker. And we give a big round of applause for the admin. Please unmute all participants. And we give a big round of applause for Nagai Sensei. Sensei, congratulations. Thank you very much. We really appreciate your great presentation. Now it's time for us to go.
Materinya, cuman kalau ininya ya.
So many boys.
Okay. Because some So please unmute again all participants from admin.
Ada.
Yes, it's very amazing presentation. And Yes, thank you very much, everyone. And admin, please also unmute Nagai Sensei and Prof. Oki. Admin, please unmute Nagai Sensei and Prof. Oki. Okay, thank you very much. So we would like to give once again, our greatest appreciation to Nagai Sensei for delivering a very impressive talk. And also, I would like to say hello to those who are joining us live, not only in Zoom, but also in YouTube. More than 100 persons are now live streaming Sensei in the YouTube channel. So many people are very excited. So in total, more than 250 participants, close to 300 are watching this talk right now. And I believe that we could get many useful insights to be applied in our research later on. And before we move on to the next one, I would like to inform that we have selected several of the questions that we think is the most relevant to the topic. And the first one, Nagai Sensei, the first question is, your presentation is very interesting, and we all think that. Thank you. And everybody is very interested about the statement, the nail that sticks out too much and go opposite with the research trend. And the question is how to formulate a good research question and how to always go opposite in formulating our research question. It is because, as we know, the question will be very important for us to catch someone's eye and also will influence our step to conduct the research. And the same person also asked for the second question, how to determine that our research problem is the good research problem. So probably this is an extension of what you have mentioned in your talk. Please, Nagai Sensei.
Okay, both question is regarding the good research problem or good research question. I think you don't think about this. Any question is okay. So the nail that sticks out too much means lots of questions. So for example, when I was a high school student, I often gave a question to the teacher, and the teacher couldn't answer to me because my question was too difficult. But kindly, my teacher asked to his previous laboratory when he was in university, and I got answer from the university. So this was quite important experience for me. So this accelerates my question to the teacher. Like this, when I was in university, so when university student, I also often ask question to the teacher, and there is a very impressive memory. So when I was a master course student, I attended the conference, scientific research conference, and I gave a poster presentation. But before poster presentation, I attended the oral presentation. And in the oral presentation, there is a plenary lecture from the very famous professor. He just came back from the US to become the full professor in the top university in Japan. And his talk was quite impressive. But there is a logical gap between them. Therefore, I asked him, although your presentation was quite nice, however, from this result to this result, there is a gap. So how do you think about this? I asked him, but he couldn't answer to this. And after that, he came to in front of my poster, and he said to me that, who are you? And which laboratory are you belonging to? And which position are you? He said to me. He didn't think I am the master course student. He thought I am the research associate or assistant professor in someone's laboratory. But it's not true. Anyway, what I want to say is many questioning is important. not to think about good question, good, not to think about searching good research problem. Among the lots of questions, there should be a good question. So that's my answer.
Okay, Tensei, thank you very much for your answer. And the second question is how to determine that the research problem is a good. Is it similar answer?
It's similar. So for example, when I was a student, there is no GFP, there is no single molecule imaging technique, but in the laboratory seminar, I introduced, we need single molecule imaging from now on. And a teacher said to me, are you kidding? No one cannot do this. How do you want to do this? But that's why I want to do, I said. But all of the teacher said to me, are you kidding? Because this is not a good research problem. So therefore, so second question is totally based on the question from others. But many people must be confident, so must believe his or her own question. His or her own question is good.
Yeah.
This is important. Don't think about others' opinion. Therefore, I said, go to the opposite side. Many people want to follow the opinion, others' opinion. Therefore, many people want to go to the famous research or other people, how to say. Anyway, another opposite side has good research, I think, I believe.
Thank you very much, Nagai Sensei. I think very clear answer. And I just want to say hello to the person who is asking this. The first question and second question is from STMIK IBBI Pak Hartono. Selamat sore, Pak Hartono. I hope if you could say something or turn on your video. Yes, I hope your question is answered. And I would like to move on to the second question, which was asked by Unsrat, Ibu Ineke. And the question is quite technical, related to your research, maybe. How do you substitute a certain amino acid in a protein and peptide molecule? It's probably more like a basic biology question.
Yeah, there is a biotechnology technique. So, for example, we can easily substitute the amino acid by utilizing the PCR-based mutagenic technique. So, PCR requires primer DNA. So usually we utilize 30 more primer, 30 base primer, and in the central, we can change the sequence of the DNA, and this encodes the amino acid to be modified, and the other part is similar or same to the target DNA sequence. By using this primer, we can substitute the certain amino acid sequence into the designed sequence. So this is a PCR-based substitution of a certain amino acid sequence in a protein and a peptide. Is it okay?
Yes, okay. Thank you very much. And the first questioner also says that you answer his question perfectly. And I think you also answer the third question. And the fourth question is from Lipi. I think you went to Lombok and you collaborated with Lipi. Mm-hmm. This is a researcher also from Lipi, but Lipi Biotech. Her name is Dr. Nilam. And she asked, what is your success story to get research ideas, and how do you get these ideas to apply to the society? Maybe you have some tips.
So the important thing is to get the research friend. And I get the very big research funding by applying the group with a group of my friend. So I often hold the dinner party, or I often go to lunch with a friend to discuss about something about the research. And by exchanging the idea or thinking the way of thinking, some people how to say, agree to my opinion. And by gathering such people, we can make the community, small community. The small community is, for example, less than 10 people. It's enough to make a research group. And in Japan, there is several grant for which the group member can apply, not individual person, group member. Among these, I often, not often, I so far tried several times. And this research funding is called Innovative Research Area from MEXT, Monbu Kagakusho in Japan. And to successfully get such big funding for group research, we must deeply discuss what we should do and what is a proper question. First of all, we have to discuss about what is our proper question, what we should solve. So for this, we need the conversation, discussion, brainstorming with several people, preferably less than 10 people, more preferably less than 6 people. So do you know Steve Jobs? Steve Jobs always held the breakfast meeting from 6 a.m. maybe, according to his book. And he said, less than 6 people is must. If there is more than 7 people, there must appear the people who doesn't say anything. Every people, attending people must say something for brainstorming. Therefore, less than 6 people is must, important. If there is 10 people, I think 3 or 4 people never say anything. This is not good situation. So therefore, for effective brainstorming or effective discussion, the less than 6 people is preferably. And from this very deep brainstorming and discussion, we can make tight community by sharing the way of thinking. And after that, we can apply the funding for group research. And after getting group research funding, we proceed the group research, and then we hold the symposium in the scientific conference to deliver our research result to the outside people of our community. So this is also important. Also, by using SNS such as Twitter or Instagram, we can spread out our research result to the worldwide. This is also important to gather the friend or to gather the collaborator. So this is our way, or my way, my method.
Okay, thank you very much, Nagai Sensei. It's very interesting. You also include the use of SNS. And I know that you are very active also in Twitter. So thank you very much for promoting our event. So I would like to again thank Nagai Sensei. And actually, we have several questions, but I'm not sure if Nagai Sensei can still answer one more question, or is it okay about the time, Sensei? It's okay. Okay. The last question, I think it's quite important for everyone also. That's why I think before I close, we can answer, is how to convince a funding agency, especially if your proposal is really out of the box and the application is sometimes still unknown. Then how do you convince? I think this is a very important question. And this is from Upi, from Judistira Arya Utama. So please share your tip as a final question.
All right. So there is several things, but the most important thing is including the preliminary data, immature data. So I'm now the reviewer of several funding, and I must read so many applications. And many applications don't include the preliminary data. Just saying, I want to do this, and after doing this experiment, I will get this result, and this result will be will contribute to the society, or will open the horizon of this research area, they say. However, there is no evidence whether their notion, their research proposal is actually achievable or not. So I always put preliminary data. So we have already obtained this data, but this is preliminary. But if you provide us funding, we can promote, we can get much more data about this preliminary data. Therefore, you have to provide me the money. So that's my way, my method. So in addition to this, the proper question is also important. Many people, for example, recently COVID is topics. COVID-19 is topics. Therefore, many people write about the COVID-19. I will make the diagnostic method, rapid diagnostic method for COVID-19 virus, or after getting the Nobel Prize in Yamanaka, Professor Yamanaka, he established how to make an iPS cell. So many Japanese moved to the iPS research, but it's boring for me. So many people, I do iPS, I do iPS, I do iPS. So this is not proper question. So the proper question is in the opposite direction. So therefore, in the research proposal, if I were reviewer, the research proposal must contain the proper question as well as the preliminary data. That 2 point is important to successfully get research funding.
Okay, Sensei, very clear and actually very important for me also. I will remember your tips. So Nagai Sensei, again, we would like to convey our utmost gratitude for your kind allocation for time for us. Then I think we learned so many things. And before I close this seminar, I would like to give you a special token of our appreciation on behalf of the ForMIND Institute. we draw for you very specially crafted for you. And if you know, this is our traditional clothes, wearing batik. And the background is in East Nusa Tenggara. It's Pulau Padar Island, which is very close to Lombok, actually. Lombok is West Nusa Tenggara. This is in East Nusa Tenggara. Very, very beautiful. And I hope you can visit this place and take this shot in your next visit to Indonesia.
Yeah.
And we would really like to say our appreciation and thank you for sharing your wisdom with the scientific communities in Indonesia. And I would like to summarize today's talk by mentioning again the key messages from Nagai Sensei to ask the proper question and make sure that you understand the word design to leave a mark. So to innovate and do breakthrough research, we have to go to the opposite stream of the current trends. So if the research that we do does not think the way that people think, then the project is a failure. I think this is a really great way to summarize everything on what you said today. And we really thank you once again. And I hope you could stay a little bit. We would like to introduce to our participants here about more about ForMIND Institute. And Sensei, this picture we will also send to you, to your office after this. Yes, I hope you like it.
Yes, I like it.
Thank you. And for the next, we would like to announce the next series under this world-class scientist. As everyone know, probably this is a series of 4 seminars. Nagai Sensei has already opened our seminar in the first series by opening our eyes and our mind. And then the next series will be more on the technical aspect on how to do the scientific writing by Professor Julian Griffin from Cambridge. And then also how to get grants. Probably it's going to be more discussion on how to get grants. That will be delivered by Professor Eriko Takano from the University of Manchester. And finally, we also have Associate Professor Jessica Lasky from Harvard University School of Medicine. She will also talk at the end of the series about how to establish yourself in the field and also how to balance women in science. So I hope Nagai Sensei also will introduce this series by YouTube, and because it's in English, everyone in your lab also is welcome to join the next series if they are interested.
Okay.
So again, I would like to move on to the next one. I hope if you haven't registered for the next series, I would like to invite you to please take a look at the link. And again, I would like to say this seminar is actually provided freely through the YouTube live stream. And more than 100 people are now presently watching live. And I hope that more people later on will be watching probably after work. But I still really appreciate that so many register. We received more than 200 registrations, in fact, and about at the peak, about 170 attended this meeting. And not only providing seminars like this from renowned scientists from all over the world, we also do many different kind of consultation, manuscript review, English editing and proofreading, project consultation, and many more. So if you are interested, we can also collaborate on providing some high value seminars such as this for your institution. And you can click s.id/jasa40. And for the next slide, please. And I would like to also mention that this is done in collaboration with Ristek BRIN. We really thank Professor Oki and Pak Adi. Pak Adi Hermanu is also here for his kind guidance. And there's also Pak Mikelio from the RPM who provide technical assistance for hosting this. And our partner, which is the Forum Peneliti Muda Indonesia, ForMIND. If you would like to take a part of it and be in our network, please register to formind.id/registration. And also you can join Ikatan Ilmuwan Indonesia Internasional in the link below. So please, next. Okay, so this is the end of the seminar. And after the seminar, we will be sending you your certificates and e-certificates by email, and also the material for the presentation today. Pak Oki, perhaps if you have some final closing for Nagai Sensei.
Thank you so much for Professor Takeharu for your very inspiring sharing and your the secret of your success story in an opposite way, in singularity, etc. This is really, really awesome. I learned also something new, and I believe all participants agree with me. So on behalf of the Ministry of Research Technology of Indonesia, I would like to thanks to you for your very kind contribution and very thoughtful as well as useful. And I believe this will be a new start because so many participants contacted me to ask whether the go opposite way will be accepted by my directorate for funding. Yes, it is acceptable because I believe, as you mentioned, as long as it is justified with a strong, previous report or some preliminary result, I think my reviewer will be more than happy to review, and possibly I will be happy also to provide funding for their new paradigm of research. Again, thank you so much. Yoroshiku onegaite shimasu.
Okay, Nagai Sensei, thank you very much, everyone. Wassalamualaikum warahmatullahi wabarakatuh. Bye-bye.
Bye.